Welcome to GIST Biomomed for your educational videos. Human embryogenesis. In humans, embryogenesis occurs in the female reproductive organ. Embryogenesis is the process by which a zygote develops into a fully formed embryo, a critical phase of early development in multicellular organisms. This process involves a series of highly complex coordinated events that transform a single cell into a structured multicellular organism. The male releases over 200 million sex cells, called the spermatozoa or sperm cells, into the female vagina during sexual intercourse. These sperm cells are made up of the head that contains the haploid genetic material, the midsection, and the tail. They swim toward the female sex cell known as the haploid ovum or egg in the female reproductive tract. The ovum is released from the ovary during ovulation. It then moves along the fallopian tube and arrive at a specific portion called the ampullar isthmic junction, where it rests for about 30 hours. The surrounding environment of the ovum produces chemicals that attract sperm cells, which can survive in the female reproductive tract for up to 5 days. As the sperm cells swims towards the ovum, the contraction of the uterus and cilia of the fallopian tube propels the sperm forward to the fallopian tube's ampullar isthmic junction. However, these rigorous journey results in the death of many sperm cells, which is nature's way of allowing only the healthiest sperm cell to fertilize the ovum. Upon reaching the ovum, the successful sperm cell first burrow through the corona radiata of the ovum. Making contact with the zona pellucida, the sperm head then binds to receptors of the zona pellucida. The acrosome of the sperm releases its stored digestive enzyme that allows the sperm to fuse with the ovum plasma membrane. These events cause the egg's plasma membrane to change composition and prevent other sperm cells from entering the egg. The sperm haploid pronucleus then enters the ovum cytoplasm, looking for the ovum's haploid pronucleus. The two pronuclei combine to form a full diploid single cell, called the zygote. This zygote stage is the earliest developmental stage that lasts for about 12 hours, and the zygote then begins its journey to the endometrium. As the single-celled zygote journeys from the fallopian tube towards the endometrium, it undergoes repeated rapid cell division through mitosis known as cleavage. This cleavage process is divided into stages of blastomere formation. Each stage lasts for 12 to 24 hours. The first stage of cleavage results in the formation of a 2-cell stage after about 30 hours of zygote formation. This 2-cell stage then further divides into a 4-cell stage after 40 hours of zygote formation. Division continues as the 4-cell further divides into 8-cell. After 3 to 4 days of zygote formation, the 16-cell stage is formed. This cell stage is also known as the morula. Finally, after the 4 to 5 days, the blastocyst is formed, which is the 32-cell stage. This blastocyst is composed of the inner cell mass, the blastocoel, the trophoblast, and the zona pellucida. During this cleavage period, the zygote remained the same size. That is, with each successive subdivision. The ratio of nuclear to cytoplasmic material increases, but the overall size of the zygote stayed the same. Note that only 20 to 30% of fertilized egg will reach blastocyst stage. On the 6th day, after zygote formation, the epiblast that eventually develops into the embryo is formed in the blastocyst. Also, the blastocyte hatches in the uterus. By disintegrating the zona pellucida and exposing the cells of the trophoblast, which then attaches itself to the endometrium, where it is implanted by the 7th day. After implantation, the blastocyst produces pregnancy hormone that tells the ovary to stop producing eggs. This marks the end of early embryogenesis. For more educational videos on embryogenesis, please like, follow, and subscribe to this channel, and also subscribe to our social media website, gistbiomolemed.com.